Hi friends, now we will study about digestion. This is the best and quality portion of this chapter. When the semi-fluid food will enter into stomach, the pyloric region and pyloric antrum of stomach will secrete some hormone like a gastrin. Due to this gastrin, when any type of hormone will secreted will mixed with blood fluid. When gastrin will secreted from the stomach, some cell will secrete some enzyme and some acid. Likewise, the HCl will be secreted by oxantic cell. Oxantic cell, I will write this. Oxantic cell will secrete HCl. It is also called a parietal cell. Parietal cell will also secrete the HCl. So HCl will be secreted by oxantic cell and parietal cell. Why HCl secreted? Because of gastrin hormone. Now another enzyme which is called a pepsinogen. Pepsinogen secreted by zymogen cell. Zymogen cell, yeah, cheap cell. Another name is cheap cell. Zymogen cell and cheap cell are responsible for secreting pepsinogen. This is inactive enzyme, and this inactive enzyme will be activated by HCl, and this will be converted into pepsin. Pepsin is active form of enzyme. Now the food particle in which protein is present will make contact with this pepsin, and now this protein digestion will start. Now how the protein digestion will start and what is protein and in which product it will be converted, we will see this in this is the protein. Protein is made up of amino acid, second strand, third strand, 4 and 5. This is protein. This is for real concept. Protein, now protein will be converted into two products, proteosis and peptone. What is proteosis and peptone? Do you know? You can search in Pradeep book, Errorless book. You will find answer. What is proteosis? The larger digestion product of protein is called a proteosis, and the smaller product is called a peptone. There were 5 chain of polypeptide in protein. Now due to digestion, it will be converted into large piece of protein and smaller piece of protein. Larger is called a proteosis. Larger is called a proteosis, and smaller is called a peptone. Now this is the topic, peptone. What is protein digestion in stomach? First you have to repeat the semi-digested fluid food when enter into stomach. Fill the topic. When enter into stomach, the pyloric antrum region will secrete hormone gastrin. Due to this gastrin, the HCl will secreted. HCl will secreted from oxantic cell and pepsinogen secreted from zymogen cell. It's also called a cheap cell. Yes. Now, in a presence of HCl, pepsinogen will converted into pepsin. This active pepsin will digest the protein. यहाँ पे हम बोल सकते हैं कि प्रोटीन का कंप्लीट डाइजेशन हो गया? नो, because protein is macromolecule and the basic molecule of protein is amino acid. You are not find that amino acid is separated in this process. When all amino acid are separated from protein, I will tell that the complete digestion of protein is completed. Now the protein is converted into proteases and peptone. What is proteases? The large subunit of digested protein. In some reference book you can also find that protein will convert into proteases and proteases will convert into peptones. But as per NCERT, I will tell you that protein will convert into proteases and peptone. Even in book it is also not written that which is large and which is smaller product. But from reference, many reference book you can say that proteases is large subunit and peptone is a smaller subunit. Now, proteases and peptone. This is not complete digestion of protein. This happen in stomach. Now, what will be in duodenum? Because proteases and peptone will enter into small intestine. Proteases and peptone will enter into small intestine. This will be converted into polypeptide. Polypeptide. Proteases peptone will convert into polypeptide. But how? And what is polypeptide? So you can see this proteases in this figure that 1, 2 and 3. 1, 2 and 3 chain are called proteases. Proteases. And these 2 chain are called peptones. During digestion of proteases and peptones, these all 3 and this 2 chain will be separated in single chain. So all 5 chain will be separated. So we will say that these are the chain of polypeptide. So polypeptide, how it was made, that you can understand proteases and peptone will convert into polypeptide. Repeat. The food when enter into stomach, pyloric region will secrete gastrin. HCl secreted by oxyntic या parietal cell. Pepsinogen secreted by Chief cell or zymogen cell. Then this pepsinogen will be activated by HCl. So this active pepsin will convert protein into proteases and peptone. This will happen in stomach. Now proteases and peptone will enter into small intestine. This will enter into U-shaped duodenum. These proteases and peptone again and again digested and will converted into single polypeptide chain. How and by which enzyme this reaction is occur? The cell zymogen and oxyntic cell are present in which layer? These are the question in your mind. This oxyntic cell and zymogen cell are present in mucosa layer. What is mucosa? Mucosa layer is the innermost layer of digestive tract. I will draw mucosa layer. Actual 4 layer of digestive wall. Outer is serosa layer. Serosa layer. Second one is muscular layer. Third one is submucosa. Third one is submucosa. And fourth one is mucosa layer. you learnt about goblet cell. Goblet cell is present in totally digestive tract, even in pharynx, even in esophagus, even in stomach, even in small intestine, even in large intestine. But these goblet cell are present in mucosa layer. As same as here, in mucosa layer, the oxantic and zymogen cell are present. This may be question and need examination. So remember this. Now, how this reaction will take place in duodenum? This is the best question, and another three answers are also based on this topic. We studied that pepsinogen is the responsible inactive enzyme in this protein digestion. Now, which one is responsible for proteolysis and peptone digestion? The answer is trypsinogen. Trypsinogen. Trypsinogen is the inactive enzyme, and this will be activated in trypsin. Trypsinogen will convert it into trypsin. Likewise, pepsinogen was converted into pepsin. As same as here, trypsinogen will convert it into trypsin. From where this trypsin will enter into duodenum? This is again next question in your mind. Trypsinogen will arrive from pancreas. This is the pancreas, and this is hepatopancreatic duct. This is pancreatic duct, but this is hepatopancreatic duct. Why it is called a hepatopancreatic? Because this is the combination of common hepatic duct, which is came from liver. So common hepatic duct and pancreatic duct will recombine to make hepatopancreatic duct. So hepatopancreatic duct या enter होगा pepsinogen. So from where this pepsinogen will enter in duodenum? It is the enzyme, inactive enzyme of pancreas. So I will write P for pancreas. Please, you have to remember P for pancreas. In this, trypsinogen convert into trypsin in presence of HCl. But in this case, trypsinogen will convert into trypsin in presence of what? The answer is enterokinase. Enterokinase. From where this enterokinase will enter? Enterokinase is the portion is coenzyme. Please mind it, it is coenzyme. This is not enzyme. This is HCl, acid. This is enzyme, but it is coenzyme. Why it's called a coenzyme? What is the meaning of coenzyme? Helper of enzyme is called a coenzyme. This is enzyme, trypsinogen. Enterokinase, it's helping to trypsinogen, so it is called a coenzyme. But from where this enterokinase will enter in duodenum? Yes, the submucosa layer of duodenum. This is serosa layer, this is muscular layer, and this is called a submucosa layer. And the submucosa layer possessing some gland which is called a Brunner gland. This is called a Brunner gland. So Brunner gland is responsible. Brunner gland is responsible for secreting the enterokinase, which is present in submucosa layer. Submucosa layer. This point is the turning point. Why turning point? Because this activated trypsin is the main functional enzyme which will performing three enzymatic activity. First one, you have been studied that proteolysis and peptone will convert into polypeptide. And the next one, this polypeptide will convert into, यहाँ पे आप देख सकते हो कि all polypeptide, this is the single strand of polypeptide, but it will be converted into smaller polypeptide. So polypeptide will converted into small polypeptide. Polypeptide will convert into small polypeptide. You can see the structure in this rough figure. Proteoses, there were 5 joined polypeptide. Proteoses converted into protein converted into proteoses and peptone. Protein, the combination of 5 chain converted into proteoses and peptone. This proteoses and peptone converted into single strand, any polypeptide. This polypeptide will converted into small polypeptide. How this reaction occur? So in this case pepsinogen, in this case trypsinogen, in this chymotrypsinogen is responsible. Chymotrypsinogen will converted into chymotrypsin for synthesis of small polypeptide. How polypeptide will convert into small polypeptide? In a presence of chymotrypsin. Chymotrypsin is made up by chymotrypsinogen. Like pepsinogen to pepsin, trypsinogen to trypsin, like same chymotrypsinogen to chymotrypsin. But in presence of HCl, in presence of enterokinase, in presence of what? So I told you that the trypsin is performing 3 reactions. This trypsin is responsible for to convert chymotrypsinogen into chymotrypsin. Another last reaction of trypsin we will study in the next. This is small polypeptide made up of 4 amino acid, 3 amino acid. Now again the digestion of protein is continued, it is not completed. It will convert into 2 amino acid, yani dipeptide, or we can say that peptide. Now we will study that small polypeptide how will convert into peptide or dipeptide. You can say what is peptide? 2 amino acid, combination of 2 amino acid is called a dipeptide or peptide. This is complete digestion? No. Next, this dipeptide will convert into amino acid, and at that time protein digestion will be completed. It is continue. How this small polypeptide will be converted into peptide or dipeptide in a presence of procarboxypeptidase. Procarboxypeptidase. When procarboxypeptidase will convert into carboxypeptidase, at that time this small polypeptide will convert into peptide or dipeptide. So again, in a presence of what? So this is the third and last step of trypsin. So in a presence of trypsin, this procarboxypeptidase convert into carboxy. This is not hard name, very simple. In stomach there was pepsinogen. In this trypsinogen, P, T, simple. Pepsinogen, trypsinogen. काइमो लिख दिया, काइमोट्रिप्सिनोजन, देन प्रोकार्बोक्सीपेप्टिडेज, सिंपली. नाउ डाइपेप्टाइड यानी टू अमीनो एसिड यानी डाइपेप्टाइड और पेप्टाइड will convert into amino acid yani single amino acid in a presence of erepsin. Please, this is the main and last, erepsin. Erepsin, another name of erepsin is called peptidase. Peptidase. Why? The erepsin is one of the best enzyme. The answer is that this is inactive and active. Inactive to active, inactive to active, inactive to active. But erepsin is already active enzyme. So this is the odd one enzyme which is related to protein digestion, because all enzyme have to be activated, but this is already in active form, erepsin. Now protein is converting into amino acid, so it is protein digestion completed in human body. Second, there are many questions. Wait, there are many questions about this. From where chymotrypsinogen will enter? So it